Hello everyone, my name is CJ, and I'm really excited today to present to you this article on the impact of entacapone on the microbiome and how iron sequestration plays a role in that. The microbiome has been suggested as the last human organ because it has profound impacts on the health of an individual, including the ability to modulate or influence the bioavailability of orally administered medication. If you want to read more about this figure is from a great paper about how drugs influence the microbiota and vice versa. The bottom line up front here is a pretty obvious one. Entacapone disrupts the human gut microbiome. Everything we ingest has the capability to alter our microbiome, and entacapone does just that. The meat of this paper is really digging into exactly how this works. So this study surrounds this big question of how do human-targeted drugs impact the microbiome, which is just a very broad and big question to ask. There's a lot of factors that go into this question, like what is the mechanism of action of the drug? What does it treat? What is the chemical structure? Does it target specific bacteria? And more. Because of this and how variable the answers can be, we often see this broken down into smaller studies, drug by drug, like we see here in the investigation of entacapone and loxapine succinate. We already know that there are big associations between the use of medication and alterations in the gut microbiome. A study cited in the introduction of this article shows that 24% of human-targeted drugs can inhibit the growth of at least one gut microbe in vitro. A big important note on this is that the authors write in their introduction that there's this idea that drugs can alter the gut microbiome and gut microbiome functions without impacting or inhibiting growth or changing taxon abundance. This means that more than growth kinetics needs to be observed in determination of pharmaceutical influence on the gut microbiome. Also, just a few more notes before we really dig in. Entacapone is a combination drug. It's administered together with the main Parkinson's treatment drug, levodopa. Entacaptone's main job is to prevent degradation of levodopa so it can do its work. Loxapine succinate is a schizophrenia treatment. So why do we care about these things? Why does this matter? Prescriptions for these drugs alone exceeds 30 million tablets in the United States as reported in this paper. These are widely used drugs and thus have wide impacts on our community health as a whole. And lastly, remember dysbiosis can be caused by a lot of things, but it also can be the cause of a lot of things. Brain disorders, like those targeted with entacapone and loxapine succinate, are very susceptible to dysbiosis. So if the medication that is used to treat these disorders also causes this dysbiosis, is the help worth the harm it does? So right off the bat here, I want to address the sample size of this portion of the study. For this experiment, the researchers collected fecal samples from healthy adults and incubated aliquots of these fecal samples with the drugs of interest, entacapone and loxapine succinate, which, by the way, I'm going to call them Ent and Lox because they're easier to pronounce and mouthfuls. So back to the sample size. Only 9 samples were collected from healthy adults, and only 6 of these samples were used in the experiments depicted in Figure 1, 2, and 3, with 2 male samples and 4 female. I would also like to mention that there's very minimal information stated as a bar for what constitutes these people as healthy. The bar is as follows: no antibiotics in the last 3 months, and no self-reported history of digestive disease. Again, this is self-reported history. I don't think that this discounts the study or the data presented here nearly at all, but I do think it's important to mention. And while we're talking about study limitations, I also wanted to throw in something that I noticed in the methods section. The fecal samples were prepared and pooled, not treated as individual replicates. They did perform the experiments in triplicate, but those are technical replicates, which are more often used to assess the fidelity of the assay itself and not the delta between individuals, which you can't do with a pooled sample. Okay, so these fecal samples were incubated with either no drug, high concentrations of a drug, or low concentrations of a drug for either 6 or 24 hours. Drug concentrations were determined in 2 ways. For the low concentration, a previous concentration from another study of theirs was used. Basically, in that study, they were investigating drug effects on pure culture isolates, not on human fecal samples. The high concentration is used to intend to mimic the concentration of the drug in the colon. So for ENT, this was based off a published reference of colon concentrations, which was crazy helpful. But for LOX, there was no data available like that. So what happened is they estimated a concentration that was used based on other published in vivo drug concentrations of drugs in this same chemical and therapeutic category. And then real quick, let's run through the functional outputs so we can dive into this data. They measured the microbial loads via flow cytometry. They measured the microbial community profile based on 16S rRNA sequencing and metagenomics to construct microbial genomes, or MAGs. And they traced microbial activity changes on a single cell level using isotopically labeled heavy water. Okay, microbial loads. They do this via flow using counting beads to determine the exact microbial loads. The bottom line of this slide is that ENT at the high, or again, the more clinically relevant colon concentration doses, majorly disrupts the microbial load after 6 hours. But this is not permanent and shows a clear attempt at recovery after 24 hours. 16S rRNA sequencing was performed via Illumina, which makes sense for a study like this, looking for the breakdown of different organisms in a sample. And since we just learned about barcoding, I thought it'd be cool to point out that that was used here as well. So we can see here that comparing the data from panel B is mirroring C and D. The absolute abundance, which is shown in D, is directly following this pattern we see in panel B of increase of microbial load for all samples over 24 hours, except for the ENT high concentration, which shows that decrease in the microbial load and the absolute abundance at 6 hours, with that attempted rebalancing effect at 24 hours. In panels C and D, we can see the breakdown of alpha diversity in these samples. C depicts the relative abundance, while D depicts the absolute abundance. And quick reminder, absolute abundance is the hard count number of each type of microbe in the sample, while relative abundance is normalized to the total sample population. And in this heat map assembled using long-read metagenomic sequencing data that shows alpha diversity in each sample, I just want to draw attention here to how ENT flattens the diversity and lights this whole column nearly up in blue, while every other sample shows an increase in alpha diversity. We see some red start to creep back in and take over again after 24 hours, but entacapone is a drug that is usually administered for multiple daily doses. So recovery after 24 hours like this is not really likely in a true patient in vivo situation, because the patient would be taking this multiple times within that 24 hours, and there would be no chance for that lag of entacapone to allow this regrowth. These results were confirmed through a repetition experiment with different media used as well. And before we get into the next set of data, I wanted to touch quickly on SRS fish. I know that most of us has probably seen fish, but I didn't know what SRS was until I read this paper, so I learned something new here. So SRS fish is a combination of two experimental techniques. I won't get into like the nitty-gritty of either, but SRS is used to detect metabolically active cells by detecting incorporation of heavy hydrogen isotypes, or heavy water. And FISH is fluorescent in situ hybridization, which is the one that I think most of us might most likely have heard of, but uses fluor-tagged complementary DNA to search for target DNA on a sample. So when it finds the target DNA, it sticks and lights up and is microscope detectable. So big important note here though. The ENT high concentration conditions. Reminder, ENT high is the more clinically relevant dose. The colon concentration is what they're trying to replicate here. The ENT high concentration conditions were excluded from this next set of assays in figure 2. The researchers mentioned that there was an unexpected high non-vibrational background that made these samples impossible to analyze, and this is discussed later. Focusing our attention on panel A, this SRS data clearly shows the addition of drug impacts the metabolic activity of the pooled fecal sample in culture. Notably, the LOX high condition is the most impactful. Unfortunately, even with the LOX high concentration showing decreased activity, we can't make assumptions on how the ENT high concentration would look. In my opinion, this figure is actually one of the coolest, and it relates back to the previous microbial abundance data. If we pull back in our absolute abundance data from figure 1, we can see that even without impacting the microbial abundance, every drug condition does impact the microbial metabolism. LOX low and high concentrations didn't show that crazy dip that ENT high did in absolute abundance or microbial load, but we still see here on the left that there is some alteration in the cells compared to the no drug control. And here we can see the fish data, which highlights specific taxa of bacteria found in the fecal samples directly compared to the SRS data to show metabolic activity. It's easy to see that the Clostridium species are very much present as targeted by the fish probe, which is in blue, and they are metabolically active as determined by the SRS image below, which is in red. It's also easy to see that the LOX high concentration has a different SRS profile than the no drug condition. Overall, there's this trend of decrease in metabolic activity in the high concentration LOX conditions. Some taxa are more sensitive to any of the drug conditions used, and some are less so. Fish data is reliant on the idea that the cells, while impacted by drug treatment, are still alive and alive enough for detection. That isn't the case with all the species analyzed here, like P. dori, which will come back later in a different figure. And the researchers provide a few reasons why the fish data itself isn't more robust for a few species due to the drug-induced death. Luckily, the fish data was quantified for us and thrown into these bubble plots so we can get a more clear overall picture of what's going on. These bubble plots are showing differences in the fold change of either metabolic activity based on SRS data or absolute abundance. So brighter or more yellow bubbles are positive fold change, meaning growth, and darker, like the purple-red bubbles, are decreases in activity or abundance. Size of the bubble relates to the p-value or the statistical significance. The bigger the bubble, that means the more significant. Super, super clear message here. All three drug treatments are causing a decrease in metabolic activity, and LOX high is the worst. Again, based on the microbial load and the abundance data from ENT high, I really wish we could have seen ENT high data here. And knowing what I know with what comes later, I know why we couldn't, but it would have been so cool to see. The abundance data is a little bit more confusing, with some taxa being positively impacted by the drug treatments and others being obliterated by them. P-values were determined using the Wald test and correlated using the Benjamini-Hochberg method. Okay, so remember that situation that I was talking about that caused us to be unable to get the ENT high treatment groups for the SRS fish experiments? They did discuss directly what caused that, and it was due to a strong, unspecific SRS signal that drowned everything else out. They did investigate this, and they found that ENT accumulates in the microbial cells, which causes a disruption called a photothermal signal, or PT for short. So, in short, succinctly, the researchers measured the PT values of entacapone itself, and then compared these values to PT values of ENT high treated cells. They found that the PT intensity of the cells was high, like the entacapone sample. They were also able to show the correlation between SRS data, again, metabolic activity, with this PT intensity, or the entacapone accumulation, seen in panel D. These are highly registering PT cells are very low in SRS, showing a negative correlation between ENT accumulation and metabolic activity. Taken with the SRS LOX data, this tracks well. So they looked at the PT signal and the other members of the microbial community that were targeted earlier in figure 2 with the fish probes, and they found varying levels of bioaccumulation in each taxa. Based on this, they also looked at the impact that the bioaccumulation would have had on growth kinetics of each of these microbial members, and again saw varying levels of responses. Some showed strong growth inhibition, and others looked rather normal or even had growth promotion, like E. coli. Okay, so we know all this stuff about the way these two drugs impact the microbial community in fecal samples, but I just wanted to run through very quickly with you all the things that we've covered so far, because this is a lot of information. ENT at estimated in vivo concentrations has a substantial negative impact on the microbial load of ex vivo gut microbiome samples. ENT also flattens the alpha diversity of these ex vivo gut microbiome samples completely. And any of the nervous system targeting drugs tested show negative impact on the metabolic activity of the gut microbial community. And lastly, entacapone bioaccumulates, and this also correlates with decreased metabolic activity of the microbial community. So how and why do these drugs inhibit microbial growth? Is there an intervention strategy here that we could use to help mitigate these impacts? Based on the chemical structure of entacapone, it was hypothesized that the drug may be chelating available iron around it. Iron is a precious and essential nutrient for gut microbiota. Decreases in the surrounding iron levels could cause negative effects in the entire gut microbiome. So, in a colorimetric test called the siderophore assay, which is used to detect small molecules that bind to iron, so in this case that would be the entacapone drug, it was clearly shown that the drug is chelating all available iron in the environment. The blue color seen in the control wells indicates there's no iron chelation occurring, thus no siderophores. The color change to pink indicates the opposite. What's interesting here is that the addition of extra iron in the form of ferric chloride shows a saturation effect based on titration. The more iron present in the environment, the less siderophore activity is present in these drug conditions, which might indicate a potential intervention if iron availability is the mechanism by which ENT reduces alpha diversity. If we remember from the SRS fish data, P. dory was severely impacted by the high concentration drugs. For this reason, it was chosen to evaluate the hypothesis of iron scarcity inducing microbial death. The culture was treated with no drug, ENT high, or ENT high complexed with different types of concentrations of iron or cupric ions. So the cupric ions were used to test the specificity to iron to see if the chelation was any metal or if it was specifically iron. And first things first, it's very clear that the specificity of entacapone is to iron and not just any metal ion. Then we can see that the complex entacapone treatments, those that are preloaded with iron and already saturated, show no inhibition of growth of this highly sensitive bacteria. And lastly, we see that there is a threshold of environmentally supplied iron that will hold the entacapone at bay and allow the growth of the bacteria. Following the experimental design of Figure 1 with the fecal samples incubated with the drug and then assessed for microbial load and diversity, we see that both treatment with pre-complexed entacapone and supplementation of extra environmental iron to saturate the administered entacapone has less impact on microbial load and rescues microbial diversity for the entire commensal gut microbe community. Here we can see the statistical significance of the changes in alpha diversity and the absolute abundance of the commensal gut bacteria when treated with entacapone with or without iron supplementation or complexing. It's pretty easy to see in panel E that the alpha diversity is closer to the control when ENT high is supplemented with iron. And again, in panel F, absolute abundance shows the major negative impact ENT high has and how iron supplementation can rescue this. In panel E, alpha diversity was evaluated using the Shannon index and the inverse Simpson index as well. And in panel F, the Wald test was used and the Benjamini-Hochberg method. Measurements of ASVs were used to determine absolute abundance, and 25 of the most abundant ASVs linked to the metagenome assembled genomes, or MAGs, from earlier experiments in Figure 1 was analyzed. NanoSIMS was used here in panels A, B, D, and E. And NanoSIMS is a mass spec technique that analyzes the elemental composition of samples, in this case the fecal samples and known ENT accumulator bacteria P. dory, were analyzed for iron and carbon concentration. These data show elevated levels of iron in cellular samples treated with ENT, suggesting that the drug-iron complex is stable, can penetrate and accumulate in cells similarly to the drug alone, and can be sustained intracellularly. But this figure is pretty quick and easy. It just confirms that the entacapone carries its complexed and chelated iron into the cells that accumulate it. But it's likely that the cells still die from iron starvation due to an inability to pull this iron back from the drug complex and use it. It is at this point that the authors make the broad statement of, these results suggest that complexation of limiting nutrient iron by entacapone is the primary mechanism causing the strong inhibitory effect of entacapone on the microbiome. There is one more figure left, but I wanted to pause here and ask if everyone agrees with the synthesis of the last 5 figures. Is it fair for the researchers to draw this conclusion at this point? Do you think they could have drawn it earlier? Or do you think further analysis was needed before they say this? So let me know. That'd be a cool discussion topic. Okay, so last figure, figure 6. What's up with the commensal bacteria that could grow in the end-high conditions? It's clear that the siderophore activity has detrimental effect on the growth of most bacteria, so why are there some strains like E. coli that could survive just fine in these conditions? Going a little out of order in the panels here, but much like they isolated the mags earlier, they were able to isolate an E. coli mag from an end-high treated sample, and when looking through the genome, they found this cluster of genes that functions to make and release siderophores. So panel A is the gene schematic of this. Panel D is their working hypothesis on how siderophore-producing bacteria are able to interact with the environment. If you're like me, you might be wondering, how exactly does this work? Producing molecules that complex with iron doesn't explain why they can thrive in the low iron environment. So just very quick aside, this article gives a great explanation on how this works, and if you're curious, I really encourage you to look into it. Essentially, the siderophores go and find the iron, and then those siderophores are trafficked back to the cell to bring them in, like little chelator nets. So in panel B, they looked at the basic growth of E. coli strain E2, either with or without both the drug and supplemental iron. It's clear here that the presence of iron itself is a main driver in growth promotion, but we can see in end-high conditions, this strain can indeed grow, unlike other strains previously seen. Quick question for everybody. Here they used an E. coli strain E2, which they state is very similar to the E. coli strain D that they isolated and identified the mag of that was in the previous slide with all the siderophore genes. Do we think this is an adequate substitute here? Is it acceptable to use this E2 strain as a model in this case instead of the D strain? The methods section have sort of some background information that might help you answer or support your answer on this. Specifically, the section isolation and sequencing of E. coli isolate might be helpful. Moving on, panel C shows some really cool genetic mutants where a wild-type E. coli strain had an enterobactin production gene or an enterobactin uptake gene deleted, and then each mutant strain was compared in growth to the wild type under different conditions. So all of them grow in the no-drug conditions, but there's a clear deleterious effect seen when grown with end-high and the subsequent rescue effect of the Ent-B mutant once exogenous enterobactin is provided. So another question for everybody, which gene deletion do we think is more impactful to the health and survival of the organism, Ent-B or Feb-A? This feels like an easy question, but I think if we really dug into the discussion of like cellular metabolism and energetics, it might be interesting to brainstorm. I have a feeling that all of us are going to come to the same conclusion, but it would be cool to see what anybody comes up with. Lastly, in panel E, we see that when a culture is treated with entacapone, it increases the AMR and virulence index. This data was synthesized by evaluating all the mags cataloged earlier in the article for AMR or virulence genes. Siderophore production is often attributed to virulent and pathogenic bacteria, and we can see that entacapone treatment selects for these bacteria that contain AMR and virulence genes, regardless of the fact that these participants have not been on antibiotics in over 3 months. Last question for all of you. Does anyone have any ideas on the mitigation of this effect? Maybe combination of probiotics, advisement of a particular diet regimen? Would love to hear any information or any thoughts on that. Yay! So we're close to the end. Here's the wrap up. Why do we care about this stuff? It's likely that not all of us are personally prescribed entacapone or loxapine succinate, but studies like this are not intended to be relevant specifically to you as a human only. They're chipping away at this larger goal that we talked about earlier of how do pharmaceutical drugs impact the microbiome. The more information we gain in response to this question, the more likely that you will end up taking a drug at some point that is covered in one of these studies, and the administration of that drug is hopefully improved based on things like this. Something we can all be sure about is that we don't want our therapies to cause more harm than good, and microbiome studies can help that. Here we can see that these drugs are more complicated than just directly inducing microbial death. I'm bringing back this figure from the article to highlight their final hypothesis, that entacapone doesn't directly mediate death in cells. It bioaccumulates in cells and complexes with iron to hinder microbial access to an important and essential nutrient. We often think of drugs that impact the microbiome similarly to the way that we think of antibiotics, that they target the microbes directly to kill them. But this study shows that pharmaceuticals are often more nuanced than that, and sometimes they're interacting with an abiotic environmental aspect that causes a ripple effect through the whole community. This can shift microbial communities and their taxonomic makeup in favor of bacteria that host virulence genes, which adds a layer of caution to their use. This slide summarizes all the questions I posed, so no one has to go searching for them. I really enjoyed this article. I hope you did too. I thought it was really interesting to see that the mechanism of alteration here relied on this abiotic environmental change rather than a direct induction of lysis or apoptosis or something similar. And here are my references. A lot of these are really good reads, so if you're interested in the intersection of microbiome health and disease treatment, this would be a cool jumping off point for you. Highlighted is the article that we discussed. Thank you all so much for your attention and your time. And I'm really looking forward to our subsequent discussion.